in biology. And so thinking about what ways we can work together where their more applied side reaches your more academic, obviously basic science research going on. And so I think it's a really good opportunity for us to learn from each other and see what ways we can work together. So I want you thinking while you're hearing people present. And some of the good things about this are we're going to have a funding call associated with this. I'm going to give you the information at the end of the day. I'll get it to anyone who is not able to stay the full time. But it's also an opportunity to really think in the future, like what's happening, what can you see on the horizon with what you're doing, but nobody is really doing it yet. How do we get there? What can we do that's something unique and big and different than what's already happening out there? So think funding sources like an ARPA-H or a DARPA or an IARPA, somebody like that. So that's what we also want you thinking about today when you present, not just narrowly on maybe one project, and maybe you're presenting on one project, but we want to see how could that relate to the bigger picture of what's happening in your field. So up first, we have Omer Enon.
Thank you. I'm guessing I should use the mic.
You can use the one on the table or that.
Oh, I'll just use this. I can't trust myself to stand still in one spot, so. Okay, perfect. Thank you. Oh, cool. Nope, that's the wrong button. All right, cool. Thank you. So thank you, everyone, and I apologize in advance. I've got to take off right after I give this talk to go to run to another meeting, but it looks like colors are kind of weird here, but that's okay. It doesn't matter. So have some disclosures. Just, we have a couple startups out of the lab, some industry-sponsored research. CardioSense, actually, I'm presenting on Sunday at American Heart Association. We're a finalist in their innovation pitch competition, so that's going to be fun. And our throat was an earlier stage thing focused on more of the joint health technologies. So I'm going to kind of talk about two areas here. I'll keep it short. I guess we have a 10-minute window here. Hopefully I'll stay within that. First is some of the work we're doing around wearable cardiac sensing. Second is some of the work we're doing around joint health monitoring. So the heart is an electrically driven mechanical pump. And many times a lot of the sensing that's done for cardiac activity is based on the electrical aspects or electrophysiology through ECG and other sorts of approaches. Many of the wearables that are out there are measuring the signal called the PPG, which is the optical measurement of blood volume pulse. Typically with the wrist, they're measuring it, but this is a classic thing that pulse ox measures from finger. So our group's been very interested actually in the mechanical pumping action of the heart and how we can measure this non-invasively through the vibrations of the body in response to the heartbeat. And so we built this device, which is, this is now kind of the basis for what CardioSense has taken and is trying to commercialize, but essentially this device has multiple modalities of cardiovascular sensing on board. It's a chest patch, similar to Zio patch for MyRhythm, but specifically goes on the sternum. It measures, of course, the electrocardiogram through two electrodes, with which it's sort of adhered to the sternal wall. And then it also has optical sensors that measure 3 different wavelengths of PPG, and it has a 3-axis measurement of seismocardiography on board. And maybe more important than the device itself, this is kind of the classical approach that we've developed. In our lab, on the company side, we're doing more of a, I would say, kind of cutting-edge AI approach to dealing with these signals. But essentially, we've had a lot of experience and practice in how to deal with these time series physiological signals and relate them to clinically relevant markers. So over the years, oh boy, that's why I just use my own computer. But that's okay. So we've done a lot of studies, mainly NIH funded, as well as Office of Naval Research in different areas with clinicians across the country. A lot of it has been focused on wedge pressure estimation with the wearables, so trying to have basically a non-invasive version of CardioMEMS, if you're familiar with that technology. The potential advantage there could be for patients with heart failure, which is where we got the FDA breakthrough designation in that application, where you could have sort of a remote monitoring device to assess volume status at home and treat them accordingly. But honestly, we're very interested also in human performance applications of this. So we've done some early stage work that's focused more on heat stress and environmental sort of stressors, how those impact, especially volume status through relative or absolute hypovolemia. And we've done quite a bit of work also on sort of autonomic sensing with these devices. Oh, I'm sorry. All right, so on the joint health side, I guess kind of transitioning to a completely different sort of application. So we've done quite a bit of work on instrumenting braces and sleeves with various sensors, specifically using the acoustic sensing as one modality, joint acoustic monitoring together with bioimpedance to assess fluid levels and edema, but also structural effects inside like meniscus tears or degraded cartilage in patients with arthritis. And so we've done a lot of work in trying to understand how these signals that we measure from the braces relate to underlying clinical information. So one of the studies that we did was in collaboration with the University of Minnesota, where they were doing a clinical trial focused on a new neuromodulation therapy for patients with rheumatoid arthritis. And essentially our device was kind of a tag-on that we used to measure clinical status of these patients at the same time as they were doing all this physical exam, blood work for something called erythrocyte sedimentation rate. And basically with the sounds together with range of motion and bioimpedance detection with the brace, we were able to classify with pretty reasonable area under the curve, high versus low disease activity as per the DAS28 ESR score, which is kind of the clinical gold standard for RA. And this work, I should mention, especially with friends here, we've done a lot of work on this collaborating also in the pediatric population with Sampath Prahalad, who's one of my favorite collaborators, actually worked together for 8 or 9 years now, and very innovative person in this area as well. Some of our more recent studies have been focused on trying to move, again, more into the performance or biomechanics space with these sensors. So we've done some work. If you think about kind of your Achilles tendon as a guitar string, and if you think about sort of plucking it to determine the stiffness level and sort of vibrating it with the vibration motor and then measuring the way the vibrations kind of lead to different modes of vibration on the tendon. We've done some work on the Achilles like that. We actually have a small kind of wearable that we built for this that we're also using to try to kind of pluck the patellar tendon. But the goal here, and especially this would be something that could be in collaboration with folks interested in, let's say, overuse injury prediction or trying to build a closed loop system around, let's say, an exo that's providing external sort of support for a limb in response to the internal forces or loading conditions of the joint. This could be an interesting approach for that. And then last, I guess I'm going to present on some of the work we've done on bioimpedance as a biomechanics sensing tool as well. So typically people have used bioimpedance for more like fluid status monitoring, cardiac applications like impedance cardiography. We've been very interested in the way that the bioimpedance at multiple frequencies through a joint changes as the person actually moves. So instead of ignoring motion as an artifact, actually we use motion and gate the bioimpedance changes to motion to see how fluids are moving around internally in the joint. And I'll just kind of, this is already published at this point, but we had really interesting study where we did a longitudinal study of this wearable bioimpedance across the muscles of the joint. And we determined that we could actually quantify the level of fatigue in the muscles with the measurements taken during the activity, and that would be predictive of the delayed onset muscle soreness 2 days later. So it's kind of an interesting approach as compared to, of course, the person themselves trying to predict the muscle soreness, which is very poor. So it'd be an interesting tool for performance optimization through kind of maximizing the training effect and minimizing the soreness and pain. So I don't know if that was 10 minutes. Hopefully it's hard to 10 minutes.
You're good.
Okay, good, perfect, great. And if there's, I don't know if we have time for questions.
Yeah, we have time for a couple of questions.
Okay,
If anybody has some.
Yeah,
It's really cool technology. So let me ask you a question. Could you look at this vibrations? Because you could imagine that when a heart starts to suffer from blood deprivation due to blockage, that its frequency patterns are changed because it's not as efficient at firing. Could you detect cardiac blockage in advance?
cardiac blockage is a very vague term. Do you mean something like essentially, like say, coronary artery disease based?
Exactly.
Yeah, like calcification.
Reduced blood flow
Yeah.
to the heart, so the heart is no longer a happy camper getting its fuel. It's partly getting its fuel.
Yeah.
And so it's kind of working. Or the case, for example, you're starting to develop an enlarged heart because your valves are leaking. You know, is a very quick and dirty, non-invasive, looking at the pattern of the beats.
Yeah, so those are both directions that people have tried to go with this vibration technology. So there's a company, Hard Force Medical, that was very interested in early detection of CAD through these sorts of approaches. There's people who've worked on mitral valve regurg assessment with these technologies. The thing is that I think the vibration signals are so much affected by the body, the anatomy of the body, the structure, even where the heart is located in the chest cavity, that I think it's very, very difficult to have accurate diagnostic accuracy. I think it's easier to do, let's say, monitoring or tracking of a person's condition. So I'd like to focus on applications like the heart failure application is a great one because the fact that the person maybe has a baseline state, you determine they're compensated or decompensated clinically at first, and then you're kind of tracking their changes after that point. In the same way with sort of hypovolemia, maybe you have a baseline for the person, then maybe they're experiencing some form of trauma or hemorrhaging on the battlefield or after a car accident or whatever, and then you can sort of track their condition after that. I would be interested in something like, for example, TAVR, so post-TAVR, maybe monitoring to determine if there's some potential leakage that's developing over time. So tying it to a procedure maybe has been more of on a sort of direction, but all of those things make sense for sure. But that would be very interesting.
Thank you.
Approach for sure.
Quick question for you regarding acoustic and vibration. I mean, I guess vibration sensing with regards to lung.
Yeah.
So if you're looking at patients, for example, who are getting better clearance, say with a new drug, cystic fibrosis, or patients who are getting their lungs repaired, let's say post-TB, what would be the types of devices or approaches that you would have already in-house?
So essentially during COVID, that knee system that has bioimpedance, multi-frequency bioimpedance and acoustics, we got a COVID supplement for NIH grant to actually repurpose it towards chest usage. And so we already deployed that system in Grady in about, I think, 15 people with CHF from admit to discharge while they were being diuresed. And of course, the fluid volume in the lungs was kind of reducing, so we could see different things there. And we have it actually, we're doing some work with Jocelyn now where we're trying to use this in kids to look at especially asthma compared to digital stethoscope, look at maybe pre and post treatment, longitudinal monitoring. So that system is ready to go. The problem is it's still kind of bulky. Like if you're thinking for kids, it probably still needs kind of a refinement and miniaturization step to make it a little bit more kind of usable in clinical settings still.
Some of these patients are used to having vests, for example, for mobilization of their secretions.
Oh.
Have maybe 2-3 minute bouts where they're using that vest that's a little bit percussive like that.
Oh, percussive, yeah.
So
that would be fine then. Oh yeah, no, that would be really cool. Actually, we were interested in this. We had a project with Hillrom that was sponsoring us. I think they have a bed that does some of these percussive therapies, and we were talking about trying to. That would be very, very cool. No, we haven't done that. The system is ready for that. I thought you were thinking like long term. Yeah. Awesome.
Great. I think that's it.
Awesome. Thank you.
So a lot of GTRI funding is military-based sort of data. Is this the need, particular for recovery? So you presented the context of arthritis, but it also used for recovery and also muscle wasting with age.
I mean, the knee was, that was driven by DARPA funding. So at first it was from a project called Warrior Web, focused more on rehabbing knee injuries. Then after that, the rheumatoid work was actually also through DARPA, through a program called Electrics, where they needed more objective measures of joint health in the context of neuromodulation. We've done a lot of work with DARPA has been great at pushing these areas for us.
Are we out of time?
Yeah.
We're out of time? Okay.
You can ask what he's got to leave. So if you have one more question to ask, that's okay.
Just anything else you could say about human performance through the DARPA work or something else? It sounded like you were just commenting on that. Are some of your publications showing some injury prediction potential or anything else like that?
Injury prediction? No,
Yeah,
that's a grant that we wrote, but no, that's injury prediction. I think would require, we would want to do a prospective. That's a direction we'd want to go for sure.
You do? Okay.
Yeah, no, that's not. We don't have that yet for sure.
All right. Thank you for being here.
Thank you.
We'll get you the rest of the information later today.
Yes, thank you. I'm sorry, guys.
You're good.
Thank you.
All right. Up next, we have Andre.
Definitely, yeah.
Oh, is after this?
All right. Get that. Mike's in place. And now. Oh, that's certainly not me, so I have to jump forward. Right, perfect. Oh, that's way. All right. Hey, got to learn which one is going forward. Kind of unusual going forward with a backward arrow. I guess it checks if I'm smart enough to actually not to forget the next 5 minutes that I'm going to talk. So I'll try to actually stick with the plan to give more time for discussion. I'll get us also maybe a little bit more on the schedule. So I'm really going to do very broad overview in this, there were 4 slides that I have to set the tone for what we do, and then maybe the questions will need more of for the conversation. So my name is Andrei Fedorov. I'm a mechanical engineering faculty member, and let me jump in kind of right to the title of my whatever mini talk. Really, as a group, we are working on the broadly micro nanofluidics technologies. It's kind of lab on a chip, or oftentimes you also refer them a lab on a chip, because the combination either kind of a portable devices that can run certain diagnostic application sensing, or also the whole kind of area of emphasis for us where we try to do imaging application. And in this case, a tip, it can be catheter tip or a tip of the laser beam, so to speak. That's what drives the process. And at the core of what we do, it's really we utilize what various focused electromechanical fields to achieve desired bioeffect. I will talk about 3 different applications: biomolecular delivery, sensing, and imaging. Again, disclosure is important. You know, there is some activity in commercialization of these efforts. I'm not going to talk about who, where, and why. If there's interest, please look it up. But kind of really give set the stage for what we do. What we realize that by tightly controlling the space domain, really focusing on the scale of individual cell, what you will see in a lot of technologies which I'll present, they really focus is working on single cell scale. So ability to work on the single cell scale and control in time, this is a typically kind of less considered or aspect of any biomicrofluidic devices, which are typically steady state type of devices. We flow things through. You actually could achieve some very unusual behavior features that can be exploited. Variety of different effects can be achieved. They're kind of listed here on this map, and the whole idea is to navigate this space in the right spot, and then kind of pull the trigger. The 3 technology I will talk about, each one has just single slide about. One is called dynamic sampling platform, or we call it DSP. This is not digital signal processing. It's the electrical engineers. This could all the time people confuse this as a poor acronym to use, but it is what it is. And the thing there is to try to do bioanalytics, looking at the secretome and metabolome monitoring for cell-based therapy manufacturing. There is some collaboration work right now ongoing with Steve there that we are working on, but this is an outcome of about 5 years research that led to development of the whole portfolio of technologies that can be used now for integration in different applications. The second technology I will talk about called PAIN. This is kind of stands for precise area introduction and targeted delivery, and this is really an attempt to deliver drug or gene into cell using mechanical electrical field, but in a very different form. Unlike a typical, you've probably seen a lot of mechanoperation types of device. You squeeze the cell and get some magic, open the pores, and then let the biomolecule diffuse there. We actually switch the paradigm completely. Let's say you have a tissue construct, And what you want to have, imagine like this laser pointer, and you go with the laser pointer and you kind of point at a particular cell and do the magic of creating pores and injecting a cargo material in the cells following a particular pattern. So essentially pattern introduction or drug or biomolecules in the cell, so you can create this very complex organoid sheet, or one can imagine actually a really medical application. The surgeon would open up a patient and essentially, let's say, introduce a chemotherapy agent at the edges of the mechanically removed tumor. The last piece is much more fundamental kind of problem called technology beam map. This is a technology that is trying to bring mass spectrometry imaging to subcellular scale. So kind of let me get right into the principles of all of these technologies. I don't talk about the details, but really in the DSP case, the idea is to put an interface between cell growth bioreactor, where cells are expanded and after, let's say, genetic modification grown until to produce a therapy, to be able to monitor or biochemical makeup that correspond to cell state by looking at extracellular proteins, secreted molecules, intracellular metabolites, smaller molecules that are responsible for cell biochemical machinery inside, and cell surface markers. And these are really kind of MEMS devices, utilize silicon microfabricated devices, which couples a variety of techniques from nanoporous membrane separation to remove salt, then solvent modification to, for example, supercharged molecules, lyse molecules, separate undesirable biomolecules from desirable biomolecules. And be able to do that with a few cells, really hundreds of cells at once, down to a single cell, on the scale of down to a single minute, and do it in a reusable multiplex format. And the sensing tool of choice for us is mass spectrometry. Untargeted, label-free, extreme sensitivity down to femtomolar concentrations. And so I kind of don't show you all the pretty pictures, but there is a variety of publications that we kind of put out there. There's intellectual properties developed, and we have a company, Enson Biotech, that actually commercializing this technology, has been pretty successful and well recognized in this space. Shifting gear to my second problem that I wanted to look at, as I kind of already gave you a little preview, delivery aspect. So many of you have heard about electroporation and mechanoporation as two alternatives to viral-based transfection of the cells, essentially an attempt to modify genetically cells to achieve a desired change in functionality. And so all of these fields, viral delivery, mechanoporation, electroporation, all of these work with extract of the cell, and then you process with some sort of microfluidic device to do the desired, to achieve desired functionality. None of them is kind of fundamentally capable of doing the pattern delivery, when you individually target different cells in the, for example, tissue, and also be able to control how large of an area you want to do. In other words, focus on the single cell. Think of this like, we call it like Microsoft a pen. You pick up a pen and draw a line of a different color. Think of a different genetic material that you deliver. Just really very fine and focus on the single cell. Or you can actually have a big brush that you actually cover the entire big areas. There are no techniques that right now are currently capable of doing these things. We've actually demonstrated now, this is just a concept of these, of technology that utilize two focus fields. It actually combines a plasma, cold plasma, that is being used, that is emanating from a small tiny capillary to produce electrically excited perturbation of cell membrane and porating the membrane for a transient period, and then using a process called electrospray. And so this kind of example, this is a plasma generator and an electrospray generator, which actually utilizes focused electric field to produce a jet down to scale about 100 nanometers. And it's like, essentially you can think of a scalpel, but fluid scalpel, that can introduce biomolecules inside of those pores. And they actually demonstrated the capability to deliver fluorescent molecules in a variety of cells. So this is still an early project, demonstrating its proof of function, so to speak, and now it's actually developing technologies to get to the level of practicality we want. The last piece, this is NIH, big NIH supported project, multi-million project over the last, this is going into fourth year, I think has 1 more year to go. This is actually to revolutionize biochemical imaging. And for those of you who are in the field of biochemical imaging, there's really outside of optical imaging where you can kind of optically look at things, and it's kind of fun obviously, and introduce maybe fluorescent molecules and see targeted where they all go. The really game changer would be to have an imaging capability that can do untargeted, label-free images on the scale of individual cells. And the game in town these days, the technology called multi, got Nobel Prize about 2002, something like this. It stands for matrix-assisted label desorption ionization. And the sensing tool is a mass spectrometer still. So when we talk about multi, we're talking about resolution approximately 50 to 100 micrometers. We are talking about multiple cells. It's a single charge molecule, so you have a limit on what kind of proteins you can fundamentally detect. The third thing is requires sample prep. You need to embed the sample into a matrix, which has all of the complications. So you cannot really work with live cells, so to speak. And the sample prep is very complex, oftentimes skew the signal that mass spectrometer can detect. So, and it does not produce any topographical imaging, but oftentimes topography of the cell dictates also their biochemical function. So the technology we are developing, and really very schematic, essentially takes advantage of electron microscope and incorporate inside of the electron microscope. So electron microscope is used for biochemical imaging of the cell. But we are able to introduce a high vacuum environment of electron microscope liquids. And these liquids, again, utilize non-electron spray, and it's a highly non-equilibrium process. They still persist in the highly subcooled, because of high evaporation, liquid state, impinge on the surface of the sample, ablate part of the sample, and then it's extracted and moves to a mass spectrometer. So you can actually produce both topological image by E-beam, and by non-electron spray, you can get biochemical images and then overlay them on top of each other. And really, the key concept here, first of all, electron spray allows you to do ultra-soft ionization. In other words, we can control the energy of the molecule by changing the electric field to a large range of levels, so you can actually almost do structural biology. I'm going to kind of stop there almost like, and then spatial resolution, and as I said, it's also biological, topological information. That's all that I have.
Okay.
Yes.
Awesome. Any questions?
Any questions? Yeah.
You don't have anything to combine this with spatial RNA-seq for proteome, like parallel section also?
It possibly would be possible. I'm not very familiar how you do RNA sequence proteomics, so I would need to learn more about this. But is it offline, or you extract the sample and then just run it offline?
No, you just take parallel section, and then you either, I make customers doing his body, his burfish. You can also do spatial transcriptomics, where you basically overlay a grid of oligos that capture small.
Oh, then it's
You pull out the sequence and you tag to the location on the image.
Oh, then it should be certainly possible to do that. Yeah, absolutely. I didn't think about that.
The expression with the chemical change, that would be awesome.
Yeah, that would be awesome. I didn't think about it. This is great. Jeff?
Fascinating, very brilliant stuff. But can I ask you a question, especially about the combination of the chemical and the mass spec image?
Yeah.
How much of the parts do you actually get? I mean, if I were divided into a grid, you have so many proteins in this position, in this place.
Yeah.
So, so what exactly comes out? Because I know with mass spec, and again, I'm naive, depending on the charge ratio, you get different things,
Correct. Yeah. So this is, so just to make sure, all we've demonstrated, just all this component kind of working together. If you were to come to me and say, can you image a sample? I would not be able to do it yet. This is a technology still being developed. So you're absolutely correct. Any mass spectrometry imaging, and there is, I mentioned one multi, another one is called basic. Depending upon charge you put on the molecules, also the ablation energy, you actually extract different molecules. So the full proteome coverage, I don't think it's realistic. But this technology, with tunability of the energy of the beam, does allow you to actually almost kind of do a separation, just like LC on the fly, that you can look at the different part, very large proteins and the smaller proteins. So I think the challenges will be more on the metabolite, on the smaller peptides.
That's actually very interesting. But can I just ask one more question?
Yes.
Go ahead.
Sorry, please, I'll ask him.
Well, just, it may be related, but in the ablation regime you're operating in, do you find you get predominantly water and then some small amounts? Do you have to kind of wait for a while until you get to the cell surface, or how?
No, typically, so if you think about the cell, because high vacuum environment, right? Once you put the cell, it immediately evaporates. It's almost kind of in this quasi-liquid ice, vitrified. Exactly, water bag. And so the liquid, when it impinges, you actually produce, it's a somewhat solvated molecules. And for mass spec, you need to desolvate. So this is one of the big challenges. So kind of they're controlling how much solvate you actually carry with the biomolecules is one of the big aspects what you'll be able to detect on the back end. At the moment, I don't have an answer for you. I realize that is an important question and it's an important aspect to incorporate. Yes.
Awesome. Thank you.
Sorry.
Go ahead.
And I get the mic back. So.
Great. There we go. Good.
Great.
You hear me okay? All right, great. Thank you for the invitation and thank you for the opportunity. So my name is Farman Speke, and I'm going to talk about our project with the National Science Foundation on biomarker sensing and related problems. So essentially, I want to first mention that also I'm presenting this in the context of microRNA sensing, that the algorithm foundations that we have also applicable for any molecular sensing and also in general any sensing of high-dimensional problems. So, but we're going to focus on in this project with microRNA sensing. It's established to be biomarkers for heart diseases and various cancers. In fact, every human cell has in the order of 1,000 microRNAs in it, and whenever a disease progresses, with the HeLa cancer, a very small subset of these 1,000 microRNAs change their concentration in dramatic ways. So they are referred as biomarkers, obviously. So the idea is that can we monitor these 1,000 different microRNAs so that they can early detect the disease, but also during the therapy, let's say radiotherapy or chemotherapy, we can monitor whether the health of the patient improves or deteriorates by studying the state of these microRNAs. So that's the idea. Clearly, if you can go to the hospitals and use expensive tests to monitor these state of the microRNAs on a regular basis, right? But those are very expensive. We are really motivated here to be able to do these tests at home, like pregnancy tests or glucose tests that people do at home by dropping blood in a kit and be able to measure the state of these microRNAs for screening the cancer, various types of cancer. That's the idea, basically. This is why they're in the level of the proof of concept. We don't have technology. So these are just algorithm solutions that we are trying to do. So the idea is that, okay, but building 1,000 different microRNA sensors is going to be very expensive. In addition to that, every sensor that you build is going to have multiple footprints from different microRNAs. You won't be able to have a very selectively monitor each individual microRNA separately. That's additional challenges. So the question we propose in this project is that can we use very small subset of biosensors, let's say 50, 60 biosensors that take a footprint from these various microRNAs, and then be able to monitor the state of these 1,000 microRNAs. So this is the problem we are working on. And the idea is that these biosensors are going to be very cheap, but then you're going to have a very back-end algorithm, sophisticated algorithm that is going to be able to measure the state of these microRNA or change in the concentration of the microRNA. So this is the block diagram that we propose to solve this problem. We are going to have a biosensor that is going to be developed by our biologists. That's going to design switches to measure the concentration microRNAs. Those are top of switches. We are going to have a density solution that I developed, which gives guidelines how to design an array of 50 or 60 microRNA biosensors. And then we are going to make measurements from the microRNAs, and then we are going to come up with a structure that describes the interdependency between microRNAs. And then we are going to incorporate that correlation among the microRNA interactions back to the our density evolution optimization framework that This is gonna go again and tell us how to design a better sensing system. And we're gonna cycle through this a couple of times until we arrive at, let's say, optimal or close to the optimal solution for our biosensor design. This is 60 of them. And then we are going to take measurements and we are going to construct a graphical model. And then using that graphical model, we can do Bayesian estimation to basically monitor the state of the change in these biomarkers. So that's the key idea. Mark Stinsky from Georgia Tech, he developed these switches to, these are RNA switches that measure microRNA concentrations. When at the absence of the microRNA, the switch looks like this RNA switch. And this RNA switch is programmed that when ribosome goes and reads this instruction, it's going to produce GFP protein that we can basically measure the intensity of the light as a way of measuring the concentration of it. In the absence of microRNA, this switch looks like a hairpin, and the ribosome cannot go and read the instruction. However, when we expose the switch with the microRNA, this microRNA goes and binds to this blue part of this switch, and it opens the switch this way so the ribosome can go and read the instruction, and protein is produced so that we can see the difference. So we already have these switches. We have built 13 of them. My collaborator has already built them. Then the idea is that how we take this library of switches. So ideally, we have to go up to 50, 60 switches of these microRNA sensors, and then we can combine these switches to make up like a biosensors of them. So each biosensor, let's say I, that I'm going to generate is going to be a combination of different types of switches in that. And then the measurement of that biosensors can be approximated by this linear, basically weighted sum of these microRNAs. So X is a vector of 1,000 microRNAs. Each entry of the vector X is corresponding to one concentration of one of those microRNAs. And these are the coefficients that relate to the binding affinity and the number of switches. And there are lots of parameters goes to these coefficients, right? There's a small subset of these vector AI that is non-zero, and the rest is going to be zero because every biosensor we are going to design is going to have only a very small subset of microRNA interacting. You can model the whole things as a matrix equation like this, where Y is going to be a vector of size 60, which is our biosensors, 1 integrated biosensor output. X is the 1,000 dimensional microRNA concentrations. A is a sensing matrix. So the idea is that having a Y, the measurement Y, we want to reconstruct the X or the changes in the X. That's the whole premise of our approach. Now, to the question we are first, at of course, level is the measurement noise. The question we are asking is that how do you optimize the sensing matrix A so that given Y, you can recover your X as accurately as possible? Actually, to design this, the intellectual merit of this work is that we use something we use in error control code in wireless systems. When we design random codes using graphs, and we come up with an optimal graph that will be designed as an optimal error correcting code for wireless. And I know this connection is very weird, but that's the beauty of this approach. We presented this problem as a graph like this. So the idea of designing a matrix A becomes the idea of designing a graph like this, where here the rectangles, well, these are corrupted, of course, some of the labels are missing. Rectangles are corresponding entry of each one rectangle per biosensor. So if you have 60 biosensor here, there's going to be 60 rectangular nodes in this graph. And then each vertex corresponds to one entry in this X. This is corresponding to concentration of microRNA number 1, concentration of microRNA number 2, and so on and so forth. So coming up with the optimal A means that how do we come up with the optimal graph like this? And there's a concept called density evolution that goes and use message passing algorithm and studying the messages that goes back and forth between this graph and come up with the optimal degree distribution that tells us how to basically what are the number of edges that each node has. By sampling that degree distribution, we can come up with the optimal graph, which will tell us what is the optimal A will look like. So that's the idea. I'm going to skip it. That kind of approach not only gives us optimal sensing matrix A, but also gives you opportunity to do preferential sensing. Let's say you have 1,000 molecules to make measurement of, and there are 200 of them that is more important for you. you want to measure them more accurately, and the other 800 ones, you have less importance on them. So we call that preferential sensing. And this is only based on synthetic data. We show that we can design our sensing metrics to also be optimal for that kind of setting, where this is the error rate for the regular sensing and for these 200 important situation. We show that by preferential sensing, we can even reduce this error rate further down to much lower value. And these are just a signal to noise ratio that we are making these studies. So, and this comes at a very little cost in terms of overall performance of these 1,000 microRNA recovery. So that's the concept we have established already. These are some measurements out of the microRNA interaction with the switches that we have designed already. This is shown for 8 different type of switches. The vertical axis is the GFP reading, the horizontal axis is the index of the microRNA. So the first bar is corresponding to no microRNA interacting. There's no microRNA exposed to switches, and then these are different microRNA. When it is in the environment, you get the reading that that switch has. These readings are in the separate measurements. They are not colored. So we just expose the switch to 1 microRNA and see what kind of concentration we see. So these measurements done in the separate environment. Then we look at the measurement of 50 microRNA in one of the biosensors. Let's say a biosensor that has equal number of switch, couple switch 1 and switch 2. When this is GFP reading again, and this is when 50 microRNA does not present, no presence of microRNA. This is when all 50 microRNA present, but in the low concentration. This is the reading we get from our biosensor. This is when 50 microRNA, out of 50 microRNA, only this particular microRNA is a high concentration, of the remaining are low concentration, and so on and so forth. So these are the readings that we got from our biosensors. So what do we do with this? We take these profiles that we have. So imagine for this preliminary stage, we have only 13 switches designed. So each row of this matrix corresponds to footprint of these 50 microRNA on that particular biosensor, on that particular switch. So the green means high value, the blue dark blue means very low value of reading, meaning that this particular microRNA does not interact with switch number 0, and so on and so forth. So this is the profile of 13 switches we have designed, my collaborator has designed. This is the metrics of the sensing metrics, the optimal sensing metrics profile that we come up with from our density evolution platform. So the idea is that using these 13 library of switches, how to come up with 13 biosensors that this profile resembles this profile as close as possible. So that is when we are going to get the best answer, right? So that's the key idea. And at this preliminary stage, really, we design 13 biosensors by this profile. This is the precision we get. When out of these 50 microRNA, we switch one of them from high to low, from low to high, and see if we can make measurements, correct recovery, and these are the results we get. So in the interest of the time, I'm going to skip over this also. So one of the key problems we are looking at, given measurements, how to come up with the interdependency between these microRNAs. This is very active research area in the community of the machine learning and biology. So, but the problem we are solving is that how to look at the low dimensional. So people solve this problem when all the 1,000 microRNA are measured directly. We are solving this. We are coming up with this graph from compressed measurements of Y. So by observing 60-dimensional observation, how to come up with this graph. This is a very interesting problem. We have results that publication on that topic. At the very end, I want to mention that we have another NSL project that has ended a couple of years ago when we were using E. coli bacteria for transmitting and receiving informations. These are my collaborator built these microfluidic devices that we hold send them microRNAs and send them bacteria and receiver bacteria. And these red dots are the molecules that send them bacteria transmit to the environment through the diffusion. They go from high concentration to low concentration. They come and those red dots enter the cell inside the cell, and they bind to their ligand receptors. These lux complex go interact with the gene, and they produce messenger RNAs that then produce light proteins that we can measure their concentration. So we can use this for transmitting bits, zero and ones by this kind of system. So yeah.
Awesome. Yep, that was good.
I'll give time for one question if anybody has one. Okay. Audio up next.
Okay. All right. Good morning. It's Hong Young. Today I'm going to talk about something that I'm doing in terms of developing biomedical devices for advancing healthcare. And for the sake of time, I'm going to deliver the overview of something that I'm doing. So if you have any questions, please email me or we can talk about that after this workshop. So this slide includes a few ongoing projects that I'm doing where we are developing soft material-based biosensors and bioelectronics. So basically, we are trying to utilize the advantages of soft materials instead of rigid materials because you can provide many different things that we could get the benefits when it comes to tissue interactions of those electronics for both implantables and wearables. Mostly it's funded by NIH and NSF, where we utilize also nano-manufacturing technologies to make, as you see, wearable devices and then implantable devices, mostly targeting to get physiological signals from the body. And we utilize those signals for disease diagnosis, therapeutics, and machine learning. For those of you who never heard about soft electronics, here I prepared a photo that shows two different devices. One widget, which is something that you guys all use these days, like wearable watches, wearable rings, and other things. These are great in terms of getting reference signals, but not great in terms of getting clinical levels of signals. So we have to do something because those widget devices are having mechanical mismatches to the target tissue, which is human skin here. When it comes to implantables, they are really bulky and rigid, so they have a lot of inflammation and other issues. So basically, we are trying to address those problems by using new materials, soft materials, biocompatible materials, and also at the same time, by taking those advantages from those materials, we can shrink the size as we see from those devices on the finger. That's a lot thinner, which is lightweight, and also you can have better contact to the skin here in this case because you can follow the morphology of the human skin. So eventually you can get better signals from the target tissue, which is the skin in this case. Today I'm going to purely talk about wearable devices. So these are a few examples that we recently published in developing those devices and get those physiological signals from human bodies. So as I said that by using those soft materials and those manufacturing technologies, we can basically develop any types of device platforms. It can be mounted, all those sensors and electronics can be mounted on a patch, or it can be mounted with a glove, or it can be integrated with a pacifier, or it can be integrated with some sort of mask. So by using those different platforms, we can target different signals from the human body. Again, these are non-invasive devices, so we are purely targeting physiological signals or electrochemical signals. Not working. Oh. Oops. Just got kicked out? All right, sorry about that. that guy? 46. Slideshow doesn't work. Oh, okay. Is it because of video?
I don't think so.
No. Function device. Go down. Go up 40. Right there. 46. One more time.
Leave it out, present on the a minute.
Let's try that. Can I use my laptop? No? Or because of the
Probably may have to.
Teams? Okay. Let's just use as it is. Right. I hope.
Maybe skip that slide.
Yeah. Close this out.
It's
Is that sharing? Share. Okay. Hope this works. All right. So one example here, based on the wearable device, something that you can see upper left corner. This is an integrated device that we can include a few electrodes, photonic sensors, accelerometers, and so on. And as you see that it's super thin and flexible. It's like a bandage, so it can be mounted basically anywhere. And here we are using this device by mounting them on a chest area. We can measure signals like electrocardiograms and other things. So hope this works. I'm not gonna make it bigger because I'm worried about crashing. So we are using Bluetooth, which is embedded as well as a circuit. And as you see that the device is very thin based on that membrane technologies. But it's as powerful as the conventional wearable devices by maintaining that very small form factor. We can have real-time data recording of cardiac signals, electrocardiograms and other things, respiration, motion analysis by using the external receiver. It can be smartphone or tablet. And by embedding machine learning algorithm, we can predict any abnormal signals in real time as well. So that's one example. And here, by using the software materials again, we can shrink the size further. So we are not only targeting other population, but also we can target pediatric population because of the advantages of this very small packaged electronics. And as you see, the entire device is as small as shown over here. And by using this device, we can replace other multi-sensors that are required by pediatricians to look at those signals from these big babies. Based on this, again, wearable wireless electronics, we can measure signals from the heart, electrocardiogram, here SDG, that's something that Omar mentioned earlier, seismocardiogram that measures heart vibration by using accelerometers. But also at the same time, we can measure blood oxygen saturation, which is PPG, based on these wearable devices. They're putting another device on the finger. The main advantage is that we can really have continuous daily recording by using one device instead of multiple devices that are typically required by conventional settings. Here it has to show something. So when it comes to sleep disorder or apnea detection, we have to utilize super complicated and messy setups shown over here, right? Polysomnography that requires more than 15 wire sensors and user needs to sleep in a sleep lab one night. Nobody wants to do that. When it comes to pediatric patients, same thing is happening. However, here we develop a new device that can tackle those issues by wearing this single device platform over the face area. We can still measure super high quality signals, including brain signal, eye motions, muscle activities, products and saturations, and so on. So by using that, we can make patients stay at home, so we can provide that portable at-home sleep monitoring. So I don't know whether you see that guy. He looks a lot more comfortable than previous people. And he's wearing that device over his face, and basically he can use his smartphone or tablet that we can give him. He can wirelessly measure those signals simultaneously, again from the brain signal to others, and then our embedded machine learning algorithm can detect sleep stages automatically, and also we can detect any sleep abnormal signals simultaneously. And we validated the performance of those devices by comparing our device with conventional polysomnography setting, which is shown over here. As you see that one of those data showing brain signals, black lines are coming from conventional PSG, red lines from our devices. And as you see that those signals are matching really well. And in terms of the accuracy, our device can show over 80% compared to the conventional PSG. So we're expanding this study further with Emory University and also Shriners Hospital in Chicago. We could utilize different sensors as well. It's not limited by only using electrode and other optical sensors, but at the same time, we can also integrate microphones that can provide us to measure heart signals and lung signals, basically detecting those sounds. But instead of conventional stethoscope or rigid digital stethoscope, our device can provide that continuous sound recording, over the course of time. And again, it can be shrink further, so we can target pediatric patients as well. And obviously, by using our wireless technology, we can hear the sound wirelessly. Also, we can record them, and also we can utilize different signal processing to target and detect abnormal signals. And also, we could, again, it's something that I showed you earlier. We can embed all those sensors and electronics into a pacifier here because we are targeting electrochemical data. Here we are specifically targeting sodium concentration and potassium concentration from saliva. That's why we use this pacifier. And for those sick babies, they are required to have multiple blood draws to measure those signals. But here we can utilize this non-invasive device to have continuous recording of sodium and potassium from patients. And these are data that are showing continuous data detection of saliva, from saliva in terms of sodium and potassium levels with really good accuracy compared to gold standard. So overall, we are doing many different things in terms of health monitoring based on wearable devices, but also implantable. So basically, we are very much open for new collaborations. So if you're interested in this research, please let me know. Thank you. Any questions?
All right. Any questions, quick questions?
Yeah.
Can you use this to prevent SIDS?
Prevent.
SIDS, sudden infant death. I mean, you're doing cardiac monitoring.
We haven't tried that. That's the goal. One of our goals based on the NIH funding. So we are trying to get there, but right now we are getting more data from patients at the moment, and hopefully our device can provide that.
And what's the battery life on these things?
So for Bluetooth devices, the one that I showed you, you can see there's a little silver thing over there. That's a rechargeable lithium polymer battery. We typically use 110 milliamp hour, which is very similar to yours. Fingernail, they can go up to 3 days, depending on depending on sampling rate and other things. But we're trying to make it a disposable device, one time use. We use it for 3 days, remove it, and try to use it.
Awesome. All right, Jeff, you're up next.
Thank you.
Thank you.
Yeah, well, I guess I'm the token scientist here. I mean, I'm surrounded by a sea of magnificent engineers, and the difference is I'm a computational biologist, so I don't build anything. And what I thought I would do today is rather than regale you with what we are doing, is to try to suggest apropos of some future directions where we could combine all our efforts to solve really big problems that I think, based upon a number of talks, including Andre's and others, we're actually poised to do. And so, okay, that's upside down. Okay, so this is the problem. I mean, this is a cell. I mean, this is actually just a single cell. You know, this is what a real cell looks like. And so the notion that you can treat these things as isolated, non-interacting Mendelian entities is probably, unfortunately, the exception rather than the rule. You've got a pile of stuff here that are interacting. And so the question is, you know, how do you understand how life works? Because ideally, like, you're a bunch of engineers, and if this was 1800 and I told you to build a bridge, you know, you build it by trial and error with iron, and it may work, or it may collapse on your head. Or even in the 1940s, you may have galloping Gertie and not consider wind forces, and it's going to have harmonic motions. So what you want to do in biology is understand the design principles of how the thing works, and then apply it. That is more likely to yield effective treatments and understanding and design of new biological entities than doing it by trial and error. So that's the philosophy which we've been adopting. Now, if you have a system which looks like this, in the beginning, you build a parts list, right? You know, you do genomics, lipidomics, the lipids, metabolomics, proteomics, transcriptomics, and you have all this stuff. And typically what you do is you have all this stuff at best across a single cell, but mostly a soup, which has got a very heterogeneous population. And if you're interested in a disease, what makes you think that every cancer cell is exactly the same and there's not a distribution? It's very heterogeneous. So you've got all this stuff. And so the question is, how can we understand how the stuff interacts in both space and time, in both normal cells and diseased cells, and then turn this around and figure out what are the gears and wheels we want to turn to fix it? You know, most drugs actually fail efficacy trials. You know, about a third fail safety, so they may cause cardiotoxicity, they may cause liver failure, but the ones that don't, actually the big bugaboo is they don't work. And, you know, 2/3 of them don't work. And there's a lot of reasons why they don't work, and this is a very complicated problem. And there are cases, for example, there was a recent paper in Nature where you had a target in a model system, you deleted the target and the chemotherapy drug still worked. So polypharmacology is probably the rule rather than the exception. And if you doubted this, look at this thing and tell me that they're not interacting. So how do they interact? Is metabolomics, proteomics, you know, and I worry about proteins, independent objects, or are they actually talking to each other and cross interactions? So this is not an uninteresting structure of a molecule. This is BCl2. This is BXL. This is a metabolite, a human metabolite. You have crystal structures, so this is not a prediction, even though you can make predictions now. Sitting across this interface is this metabolite. Now, if it sits across the interface, this thing, and then I can just see the little head of it buried, I just superimpose these, okay, is going to disrupt an interaction. So what you have is a metabolite can, in principle, disrupt the protein-protein interaction. So now you have a physical example of coupling of metabolomics and proteomics. Now, this is an antagonist. If you actually look at this region here and you look at the odds ratio in humans, where is the second highest odds ratio of disease-associated variations after loss of enzyme function? It's actually over here. If I stick a metabolite here, it's like a piece of superglue, so it can shift the equilibrium from a weak intermolecular interaction to a strong one. So the notion that metabolites are only just enzymes is clearly just, I mean, why would just, they're the workhorses of the cell, among other things. Why would they be independent? So one has to actually look at the union of metabolomics and interactomics. And what I can tell you is in unpublished work, if you actually look at metabolites that don't interact, say interact here and don't interact here, because some of them are very promiscuous, okay? And you look at the probability that two proteins that bind the same metabolite are sitting in the same pathway, The p-value of that for 0.05, more than half of them actually couple proteins in the same pathway, even if their sequence identity is less than 30%. So this is not the methyl-ethyl-propyl series of proteins. They're actually diverse functions. It can couple pathways. So you have, my son came up with this idea like quantum entanglement, metabolic entanglement. So this is the background to what I'm about to propose. And then there's another thing. You talk about cancers, okay? 12% of cancers are caused by 2 viruses. Actually, there's a whole bunch of viruses that are oncogenic. You know about those, herpes, HPV, you know, kind of. But there's also a bunch of viruses that are oncolytic. They kill cancers. And actually, bacteria are known to cause cancers. Bacteria are known to prevent cancers. So actually, one of, I would suggest, the key players in complex diseases, and if you doubted it, look at COVID. It can cause a whole bunch of downstream effects long after the initial viral infection is gone. So this is a very simple example of an epigenetic effect. So what am I proposing? I mean, I thought perhaps this is why I didn't talk in detail, but this is what I've been thinking of, and this is kind of echoing a little bit what Andre said. We really need a multi-omics approach. It's not enough, by the way, to just simply do an inventory of who's interacting with whom. You also need to do it in space and time. Let me give you an example. The localization-based diseases, if you actually do a Google in the literature, there's a little bit of this. So let me consider the following thing. I have a protein that is absolutely normal, but the transcription factor is downregulated. So it's not in the right place. So you do the genome analysis, life is wonderful, it's perfect, it's not a plague. There's only one problem, there's a complete loss of function because it's not where it counts. And this is why I was very excited about looking at, this was the origin of my question, could you tell where they are? Because it's not enough to say it's there, because if it's upregulated here and downregulated here or missing, the average is normal. So you're completely missing a molecular component of what's going on. Now this is very complicated. So what I'm suggesting is, and obviously if you build this massive data set, how do you use it, integrate it, train what, which is now where we play, because you guys can measure it, and I couldn't measure my way out of a paper bag, is how you then convert this into predictions of understanding of what constitutes a normal cell, because what you typically see is, and there's huge variations in concentration, there's a lot of biochemical buffering. And if you doubt that, look at your metabolome before and after you eat lunch. You don't die because you had a roast beef sandwich, okay? Levels change dramatically. On the other hand, other things are very fragile. How do you figure out the fragile pieces from the non-fragile pieces of life? There's regions of stability and then all hell breaks loose. It's a very highly nonlinear system. This is where AI can play, because nowadays you can put all these nonlinear effects, if you have big enough data sets. You don't have big enough data sets, you're going to memorize the data, and all you're going to do is have garbage in and garbage out. So what I'm proposing is, and you guys are perfectly, that we embark on a nice R-page project to try to do a spatial and temporal molecular inventory, then use this information, this is not original, to actually understand what are the drivers of a disease. Pick a simple one, pick a relatively simple cancer, prove that this is what's going on, and then how do we regulate it and control it? So that was the idea. I thought I was supposed to talk about ideas rather than, I could regale you what we've been up to, but this is the distillation of about 20 years of work. So thank you.
Yeah. Anyone have any questions? Thoughts?
Since we're talking about ideas, so oftentimes if the system is complex, we have the cells already operating, and watching the cell operate is like watching the computer do its job. So we also see the same, you mentioned quantum mechanics, we see the same things in quantum calculations, right? Where they're now trying to propose these quantum computers to compute themselves. Quantum physics. So where do you stand on the experimental versus, like you watch the cell forever and all the chemicals, and you'll then have a giant training set to train your algorithm. Where do you stand on managing the complexity of this problem between empirical and
okay? So I stand on, in the art of the real world, you start off with relatively small training sets. Ideally, what you want is to do deep learning and large language models, or that's. They have millions of parameters, and if you have tiny data sets, training on them is a guarantee for garbage in and garbage out. They don't generalize. There's too little data for two things, and there are other techniques. So what I could argue is as you start to collect this data, you use various smart models, boosted random forest, the more traditional, because they deal with smaller data sets and can generalize. And then you let this thing loose. Pick problems where you can begin to look at it. For example, I think metabolic entanglement is going to prove to be very important with a lot of diseases. And if you doubt that metabolic diseases are relevant, have you heard of scurvy? It's a vitamin C deficiency. That's why Brits are called limeys, because they didn't have limes, they didn't have vitamin C, and all of a sudden they had all this bleeding and their teeth started falling out and everything. A lot of things, I believe, have to be coupled. And so start with something simple, work on a relatively simple model system as a proof of principle. You don't do the most complicated thing first, like a low-hanging fruit. Build it up, prove that these ideas have legs, and then generalize. So it's like 1, 2, 3, infinity. You got to crawl before you can walk. So the answer is, ultimately, I'd like to know space and time of everything. That's probably impractical. Okay? But you can start with, it's the art of the doable, okay? So I'm very utilitarian. You give us the training set, there's a whole bunch of other people here. We'll figure out what it's useful for. Yes.
If you think, when you think about a real large problem like this, where you want to attack it in a grand way, as you say, you may want to narrow down and pick something.
Yep.
Have you thought any about inflammation and inflammatory processes?
Oh, absolutely. You're absolutely right. So inflammation is the root of many evils, but actually it's one of a number of processes. It's also energy transduction. We're actually building kind of a unified theory of diseases. And the reason this is not nuts is, familiar with comorbid diseases? These are diseases which tend to co-occur. The question is why? Our hypothesis is, and we can predict them with a precision of about 80%, is there are common driver proteins. It's almost like an evolutionary tree. There's common driver things, and then they speciate depending upon the tissues. Inflammation is an example. Actually, energy is really important. Mismanagement of how the cells generate energy. Cancer is a classic example, the Warburg effect versus normal glycolysis. So you have to look, there's a lot of these processes, but if you can kind of understand how they're all interrelated, you can generate hypotheses. And this is what the AI tools at the state of the art can do now, even though they're imperfect, and I'm not claiming I walk on water because I don't, this is frozen, okay, is they can tell you a short number of experiments to do to either validate or refute the ideas. And so you can get, so you don't have to do 1 million experiments. You can do typically, like, for example, 1 million screening. We can find good nanomolar hits when we screen 30 molecules in an online library now. It's not thousands, okay? I couldn't tell you one, but I could give you 30 with a probability. So that's kind of how I would envision this working. But you're right, inflammation is really important and causes a lot of things.
And there are probably even mental illnesses that are neuroinflammation.
Oh, absolutely. But actually you want to know where you look. If you want to find diseases, look into a person's eyeball. Turns out that I know this was crazy, and I started looking at this when this first came up. I thought, oh my God, this is a bunch of false positives. I'm going to go kill myself, right? But if you actually look, there's a lot of ocular diseases, and there are people dying. You could look at, for example, a very simple one. If you want to know whether you're likely to have diabetes when you have your eye checked, ask if the blood vessels that are attached to the retina are leaking. It's the earliest symbol of diabetes, actually. And there's a whole bunch of other diseases that turn out to be strongly comorbid with ocular diseases. So if you want to actually look at a place where you want to know, where a lot of diseases occur, look in the eyeball. And it's easy, because it's really non-invasive, right? I don't have to go cutting you open or taking samples. Unless it's on the surface, look in the eye, okay? So yeah, and this is a consequence of the common driver factors, which it's malfunctioning, and then you can say, okay, well, I got this. So for example, you have hyperthyroidism, you have a high odds ratio of developing diabetes relative to random. Do you know if you get the flu, you have 6 times the odds ratio within a week if you're over the age of 60 of developing a heart attack?
All right.
As an example. Thank you.
Thank you.
I'm generally pretty loud.
I don't know.
I'm generally pretty loud, so I'm not sure I need the mic at all. Well, you guys were talking about inflammation, so this is where we're going. I'm an engineer.
Online, there's people online.
Okay, so then let me make sure I put this. Okay. All right. So we've just been talking about inflammation as the root of all ills. And the main
I mean, I would say one of the main cells that we're interested in my lab, we do a lot of innate immunity in human diseases, the macrophage. So what's cool about these cells is that they have two origins. They come from the fetal liver, and they migrate into all tissues, right? And they stay for the lifetime of the individual. They can also come from the bone marrow postnatally. And when these cells are being consumed or they're tired, they can be replaced by monocytes that come from the bone marrow, migrate into the tissue, get educated there, and do their best job are trying to impersonate the cells that came early in development. And one organ in which these macrophages, these dual origin macrophages are really important is the lung. But you can look at this slide here from really nice review published last month. These macrophages are basically our sentinels to look at stress and any type of stress, mechanical stress, infectious stress, environmental stress, metabolic stress, aging-related stress. All these stressors are being sensed by these cells. And what they do generally is that they talk with the tissue. So they are our soft biosensors, but they're already embedded in our tissues, okay? And what they do is they can call for reinforcement if they need to increase scavenging or if they need to organize a response. So they are the cells also that regulate the handoff of the information about the stressor to the adaptive system, the T cells and the B cells, okay? So they're absolutely essential. And we know that many, many diseases are being linked to dysfunction of macrophages. We know also that macrophages can be used as sensors of diseases, even if they're not sort of mechanistically involved in them, okay? And we were just talking about comorbidities and inflammation being, again, the cause of all ills. Well, a huge issue you have when diseases get chronicized is that you chronically mess with bone marrow development because you need to get more of these stress-related cells into the tissues. And that leads to systemic complications, and then you weaken other organs that were originally normal. So I would say these cells are really essential, and what's really important to know is that they are imprinted by the specific microenvironment that they get in when they go into a tissue. So it's not sufficient to take blood monocytes and figure out what's wrong with them. You actually need to take the cells that are in the tissue and figure out what's wrong with them. What we discovered in my lab is these cells and also their cousins, the neutrophils and other granulocytes, have a hypercondensed chromatin when they come into the blood. But when they get into a tissue, they decondense that chromatin explosively and they become highly transcriptionally adaptive. Okay? So the key is, well, how do you take these cells and condition them to be responsive to tissue cues? How can you conduct mechanistic studies on these cells? So the lung is an interesting organ to look at because it's a major entry point for pathogens, environmental toxins. So when you talk about, scourges of humanity, TB is one that has killed most, more humans in history than any other disease and still does. CF is a disease I'm looking at very closely in my lab. COPD is going to be the main killer in the world by 2025. Pneumonia by viruses, but also exposure to allergens, cigarette smoke, vaping additives, nanoparticles, nanotubes, etc., etc. Well, the main issue we're having when we're trying to study these cells is that because these cells are critical to stress responses, they've been under high evolutionary pressure, looking at different animal species, but looking also at different human subgroups. So they're highly divergent genetically, epigenetically, and Greg's lung macrophages and my lung macrophages are not the same, and that we know. So we have difficulties using animal models. We have also difficulties getting cell lines, and generally what we call those blood monocyte-derived macrophages. You take a blood monocyte, stick it on plastic, starts to grow, looks like a macrophage. It's not a macrophage. But this is what we've been using for 35 years in biomedicine to try to emulate what's happening in the lungs. So what's our solution? What we do in my lab, something very simple, okay? we realized that we can develop an epithelial cell at air liquid interface to emulate what's, the surface of the lung. And we engineered a matrix here that looks like the underneath of the epithelium in tissues. So it allows the cells for the cells to congregate, and then we can put some stressors on the apical side, so we flip this thing over. Obviously, this thing will be multiplexed in wells. So we have a couple of patents on it, including one to do high throughput development of this with Shuta Kayama here at Tech. And we can take blood cells from anybody, put them into that contraption, and let them migrate through. And the breakthrough in our lab has been to show that this mere 15-minute biological process of a cell coming from blood, traversing the biological barrier, getting to the other side is necessary and sufficient to induce the chromatin derepression. And then when these cells are exposed to whatever cues are on the other side, they respond. Okay? So we've been able, with this contraption, to show that we can emulate the adaptation and the function of neutrophils that are present in patients with COPD and CF and severe asthma. And I listed a bunch of papers here over the last 5 years. We've been extremely active in showing that the neutrophils from the patient's lungs are perfectly emulated by blood neutrophils that we can migrate in vitro in our system, provided we put fluid here. That's the fluid that comes from the lung of the patients. Okay? So we just educate these cells to look like the ones we have in the lung. But what we've done very recently is we've applied that to macrophages as well. Thinking from the biological standpoint that tissue macrophages can be replaced by blood monocytes, they're both inflammatory monocytes that then turn into macrophages, we've done exactly the same idea. We've taken blood monocytes, transmigrated them, and hit them with differentiation factors. And lo and behold, what we have now are macrophages that much better than blood monocyte-derived macrophages, have a large size, they have a bunch of scavenger receptors, they have class 2, so they can pass on antigens, and their secretome look exactly like the one we have from the macrophages we pick up from the lung of human beings. Okay, so what's the opportunity here? And we've been doing this with neutrophils, and within 5 years, we have multiple patents, multiple grants in multiple diseases. And now you're looking at the macrophages, and what they allow you to do, this contraption allows you to basically mass produce cells that come from given individuals and do personalized testing of drugs. So we're talking about genomic differences, but there's also epigenetic differences that occur when you've been exposed to pollutants, for example, or as we age, our macrophages are different. We can do multiple sets of exposure models. So obviously right now we're already funding for work on CF. We have both NIH and CF Foundation funding. We've been talking to our colleagues at the Primate Center to do BSL-3 experiments to expose these cells to TB, because there's, believe it or not, no model of human lung macrophage infection by TB. And then we're also working with the Shuta Kayama and currently, to expose these cells to vaping products. But downstream, we can take these cells and we can CRISPR screen them. We can also transfer genes to them, expand them, and potentially reinfuse them in patients. So I'm just throwing out, a couple of ideas there, and I'm open for question and also for further discussion with everyone. Thank you.
Thank you. All right. Does anyone have any questions?
Yes.
High-level question, but knowing that the macrophages consume these, I guess, invading or pathogenic cells, and then they process them and analyze them and that sort of thing. About being inspired by the biology. How much of this is analogous to the analytical process flow that we try to do in the lab, and do you think there's much more room to learn from how the biology processes these complex media? It sounds like you're starting to adapt laboratory processes to do what the biology is doing. Is that fair to say?
Yeah, I would say we do. We do the analogies like both ways, right? So one of the things that we're really interested in is how these cells process information in tissues. to be the main sentinel and interpret stressors and figure out whether they can deal with the stressors, whether they have to bring cells from the outside to help them temporarily. And generally, they allow these cells to take over for a while, and then these cells will contract by apoptosis, and then the tissue macrophage regains control. Okay, so a lot of this is happening through secretion of extracellular vesicles. So my lab does a lot of extracellular vesicle-based biomarker development, but we also engineer, extracellular vesicles to contain proper regulatory RNAs to actually put in, exogenous signals into a system when we think the system is not resolving. So we see that, for example, in babies with cystic fibrosis, they have normal lung, normal macrophages to start with, and at some point, there is an abnormal stress response where the macrophages fail and form neutrophils. And the neutrophils take over, and then neutrophil inflammation becomes chronicized. So to be able to do in the lab is reverse engineer that stage of disease where we actually force the neutrophils to give the custody of the tissue back to the lung macrophage, right? And that's by engineering extracellular vesicles, right? So a lot of it, my lab is driven by the analysis of patient samples. And then we try to recreate. So we go, top down from the biology to the engineering platform. And then we do bottom up studies where we try to reverse engineer biological processes by doing the mechanistic studies in the models.
This is like a complicated organization structure with teamwork among these different entities. You're analyzing that, and when it's dysfunctional, you go in and try to refunctionalize it.
You got it. Exactly right.
Thank you. All right. Last, just one more slide. Stephen, I think you were the one who was going to present this.
Sure. So, so at GTRI, one of the reasons for these workshops. Thank you. One of the reasons for these workshops is to try to bring together, campus folks with GTRI folks in order to do these joint funding efforts. So we started having yearly calls for this internal funding. I guess now it's 3 years ago or so, and the next call is going to be coming out. So the call for proposals, I believe, has come out or will come out.
We're announcing it now.
Okay,
and we will send out a piece of paper or an email with more details.
And the proposals are due actually in about a month. The proposals should be inclusive of both GTRI and campus folks to have the largest chance of success. There is no written requirement for that, but I can tell you that if you have collaboration between different laboratories or different campus departments, you're going to have a higher likelihood of successful proposal submission. And they should also be something that is, what we want to have is how can we get Georgia Tech as a whole and GTRI more involved in this intersection of biology and engineering. So we're looking for broad types of ideas that have multiple applications. Having applications that are both DoD focused and also just state of Georgia or clinical focus are both appropriate. But I think that, again, to have the highest likelihood of success, it's to have more broad application than just taking a very niche thing. Now, that's not to say if you have a niche application that has extremely high impact, it won't have an opportunity. But again, I think broader is better. And the size of these proposals, we are looking at both 1-year seedlings. And seedlings tend to be on the order of 100 to 150K for a single year. We're also looking for full proposals that go the duration of our normal IRAD cycle, and that's a 3-year, up to 3-year project, and anywhere from 300 to 400K per year of funding. And that is, it is at the overhead rates of state kind of funding as opposed to sponsored funding. So, 300 or 400K a year goes a long way. Anything you wanted to add?
The timeline is probably most important.
Steve, yeah,
Sure.
You said proposal due in a month,
There-
in 3 months.
Up here, it's
February 12th. Yeah, so it's going to be due in the next year,
I'm sorry.
not December. So you have time to get together with people. We're going to have another one of these workshops. You can see January 22nd. Tell your friends. If there's somebody who wants to present, let us know. But this time should really be used to get to know each other, understand what you're doing, and try to go ahead and start putting some teams together to get something down on paper for a proposal. We would love to see all of you submit really fun and interesting ideas. And I know we are at time, so I want to thank everyone. And I'm sorry that we kind of got to ask questions as we went along as opposed to having discussion afterwards. Yes.
And just one more thing, these proposals are a low lift, right? They're not very long. And if you have ideas that you're just starting to incubate and you want to get feedback on those early, please send them to us and we can take a look at them and give you feedback as to try to direct these into successful areas. This is something that we want to all succeed together with, right? So we want to get the most innovative and inclusive proposals that we can.
So I have a quick question.
Yeah.
So the last one of these was like August, September timeframe. Was there like an announcement of who actually won the season contracts?
we did, and now I can't remember who did. Do you remember who won?
I don't have the
Do
list.
you remember, Rebecca?
Nick Geis has one, the Cipher Lab. I can.
Yeah, Malad has one, and the sensor blue was.
Yeah, I think there were 3 awards from the last
reason why I mentioned
round.
that is just being able to see some of the past success stories would be really helpful as we're sort of developing the proposals going forward.
Yep, we can send that out for everybody who registered here today.
I think that's both true. And as we go 3 and plus more years in, I think we are looking to kind of find more comprehensive or larger, more significant efforts versus kind of more widespread singular one-offs. We're probably looking for larger collaborative.
Yeah.
I think that also leads to successful outcomes. Like we just kind of reflect on CMAT. Think of CMAT and came on board. It starts with this Marcus Foundation, and Chris was instrumental in this kind of having a vision to give 3-year grant. So we were actually beneficiary of this DSP proposal. And whatever we started the first project, and that was the core project that solved in schema. So this really was a single slide that was presented to the entire NSF team. So this technology will bring this capability that currently doesn't exist. And it was 3 years, and then it's like $250K a year around way to do that. So I think that vision is very fruitful.
Yes.
To get really something important.
Yeah. Definitely interdisciplinary. I mean, if 4 of you all today had an idea that we wanted to go with together, I think that would be a success, right?
So thank you all very much for attending. Oh, yes.
It's actually, discussing. So what's the best way for someone like me to reach out to GTRI and find a partner?
If you want to do this, everybody should have Trina's email. She's emailed you several times, probably put the calendar invite on your calendar. Email her, or if you know me, email me, and I will make sure to get it to Stephen or Rebecca or Ryan, and we'll make sure that we can put everybody together. So just reach out to me or Trina and we'll go from there.
And we're all happy to play matchmaker, so we'll try to find somebody that has interests that are related to the areas.
Yeah. All right. Thank you, everyone.
That's very exciting.
I think it is. Actually, we've got the tools to start. Actually, well, we are not yet fully there. They take a little extra. That's right. I mean, yes. The course going on. You should be in the. So that's, I just want to make sure that there's still playing about.
So I mean, we're running experiments every couple of weeks.
This time is great. And we need, part of the thing is to develop the tools. Because if the tools exist, then everyone would be doing it. I mean, it's the chicken and the egg.